talking about really how you don't really own the land that you are presently on. And I'm going to talk about how you don't really own the property that's on that land, the actual items that you think you own. This case, we're talking about livestock in particular. So next slide, please. Because I'm with the University of British Columbia, I am required to give a conflict of interest disclosure. I am the major shareholder of Connectus Bioinformatics Corporation, and I might mention a little bit of the work that we're doing in that. Next slide, please. Now, there's going to be a little bit of science here, but I'll keep it, pretty digestible. I just want to make a few key points. This is the influenza virus. It's a tiny, tiny virus. It's about what we say 0.08 to 0.12 micrometers in size, in the diameter. It's a circular virus. It's actually very similar to SARS-CoV-2 virus. It's just very slightly smaller. And just to put that in perspective, again, we're talking about 0.15 microns. The masks that we've been told to wear, they're 35 microns holes. So the equivalent is like mosquitoes going through a chain link fence. Okay, so just want to bring that out. And the thing about this virus is we have had it for years, different types of influenza viruses, not this particular one I'm going to talk about tonight, the H5N1, but usually if you have a viral infection of influenza, you recover in 3 days to 2 weeks. And it's not that lethal, except if you're extremely elderly and you have comorbidities. Okay, next slide, please. So it's a small virus, and it's a very simple virus. Its genome only encodes 10 genes. A gene is the blueprint to make a protein. So it makes 10 proteins. And this genome, being very simple, and it's very well studied, we know the genome sequence of it. What that means is that we know exactly the nucleotides. There's 4 nucleotides that are used in all genes for all animals and plants. And they specify the amino acids that are found in the proteins. So the gene is the blueprint to make a protein. So it has 10 different proteins. You have over 20,000 different proteins in your own genome that you use in your bodies. Okay, so this is the structure, and what's important to understand here is that most of those proteins are not in the virus particle. Many of those proteins are made once the virus gets into your cells. Viruses can't reproduce without a host. You have to enter into cells, and then they kind of hijack the machinery inside of cells that's normally used to make more cells. They use it to make more virus particles. And those proteins that are on the surface, as we can see here, only about 3 of them are on the surface. These are important for the entry and the exit from cells. So, and of those proteins, if you develop antibodies, you'll make them against all the different proteins that are produced by the virus. However, only the ones that interact with the outside of the virus particle actually provide you with any protection. Next slide, please. So one of those outside proteins is the hemagglutinin protein, the H protein. So when we talk about H5N1, we're talking about one of the H proteins that are known. There's from 1 to 16 different H proteins that have been described in influenza virus that affect different types of animal species. And we also have neuraminidase. That's the N protein. So when we say H5N1, we're talking about the hemagglutinin 5 isoform, and we're talking about the neuraminidase, basically 1 isoform. Next slide, please. So the first real big pandemic that we have documentation on is the influenza pandemic. from 1918, the Spanish flu. And what we know with that is it may have killed up to 50 million people at the end of World War I. In fact, more people died from the influenza pandemic than from the actual war itself. But in the trenches, the soldiers got it. And then when they went back to their countries, they spread the virus to all their countries around the world. And of course, after you've gone through a devastating war, there's starvation, there's high stress. All of these factors contribute to making this a more deadly virus. And it was at that time. And that was the H1N1 virus. And H1N1 has reappeared, but H5N1, the one that affects birds, was actually first described in Scotland in chickens in 1959. And actually, since then, over 65 years, we've had no outbreak of an H5N1 pandemic in human or animal populations, apart from in birds, spread in the wild. So in 2021, we actually had this H5N1 variant that was highly pathogenic. Now, to be highly pathogenic, that means that if you have a flock of chickens, that you expect at least 75% of those chickens to die. And typically it's around 85% of the birds. So this is a little bit different. And this seemed to come to North America from first being described in China around the end of 2021. And this particular variant has spread throughout the wild bird population and has killed many of the wild birds, but not all of them, obviously. Next slide, please. And so it's spread through all of these different animals. But in mammals, the virus is not optimally evolved to infect mammals. So when it infects animals and people, it's actually generally a very mild disease, relatively. It's not lethal, and the animals will recover, but it will spread. Next slide, please. So, I want to emphasize with this slide, ostriches are not chickens, and they're not ducks. They can be over 9 feet tall and up to even 350 pounds. And they are deadly in the wild. Nothing messes with an ostrich. Even lions don't mess with ostriches because they're like velociraptors. Their two-toed clawed feet can basically gut a lion with the right. Okay, so this is one of the things I found fascinating, that these birds, and we'll hear later from Dave Bielinski, who's actually handled these animals. It takes a long time for the animals to actually get comfortable with who's taking care of them. So when you have these guys coming out in white suits, I think this would be rather stressful for these birds. But actually, those people in the white suits would be much more actually vulnerable themselves. And in fact, this is one of the things that kind of makes me think that those birds were actually highly drugged to be docile.
Yes.
Okay, so these can last a long time. They can live up to 65 years. 55 years of them, they can actually produce eggs. Next slide, please. And these eggs are the largest eggs on the planet. And they are full in the egg yolk with antibodies, which is there to protect the new hatched bird until it can actually improve its own immune system. And these antibodies, though, from the ostriches are particularly interesting because you can almost boil these antibodies and they still work. This isn't true for example, chickens and ducks and other animals. And they also take a wide range of pH from very acidic conditions to very basic conditions. And what that means is these can be produced in large amounts and they can be used for industrial applications because of how stable they are. Next slide, please. So the Universal Ostrich Farms, UF for short, these, it was really founded by Dave Bielinski and Karen Epperson. And they've been trying to make a go of this with the ostrich. The industry itself has gone through a lot of tough times over the years, but there was a lot of potential for it. And if you were buying birds originally, these ostriches, especially for breeding, you could be spending easily $10,000 to $20,000 a bird. This is something to keep in mind when the CFIA is offering $3,000 a bird for compensation. So they're originally were looking at the meat industry, but really in the last 5 years, the intent really was to look at other opportunities, in particular using the antibodies that are in the egg yolks. Next slide. And so these were originally investigated for opportunities to treat acne. Antibodies can target digestive enzymes in the guts that can lower the production of fats into the body or of sugar. So they can actually be used for diabetic and arterial sclerosis potential for treatment. They can be used in cosmetics and toothpaste. So that was one opportunity. Next slide, please. But one of the things that was being investigated before the outbreak actually happened of the H5N1 at the farm was to actually working with collaborators in Japan. Dr. Tsukamoto at the prefect in Kyoto. Basically, he was using ostriches and making antibodies against the SARS-CoV-2 virus. And then when we take these antibodies and you coat masks, he proved that you could actually capture the virus on the masks. So even though masks don't normally work, when you actually have the antibodies embedded in the mask, the antibodies capture the virus as it's going through the masks. And then you can prove, if you take the mask, you can visualize the actual SARS-CoV-2 spike protein on the masks. So imagine you had someone with a mask that are coated with the antibodies against the H5 protein of the influenza virus. It would actually capture the virus in the air, because that's how you get influenza virus. You don't get it from touching surfaces. It's airborne, and it has to be an aerosol. It's very sensitive to drying out. So, and SARS-CoV-2 is very, very similar. Now, you can also take that and you can put it onto filters that would be in air systems that could clean the air to reduce the risk of actually spreading the virus. Next slide, please. So the Ministry of Agriculture actually put out $2.5 million fund to try to find a way to reduce the occurrence of the the H5N1 virus in the poultry and farms. Here was actually part of the solution. And again, in the Kyoto prefect, that's where Dr. Tsukamoto was actually working on this and showed that it actually does work. Next slide, please. So the outbreak happened between December 11th in 2024 and January the 14th. During that time, they had around 450 ostriches and 69 of them died. And Dave will talk maybe a little bit more about this. But of those that died, the birds that have been on the site already after an outbreak where there was an illness that was diagnosed as Pseudomonas, a bacteria, they had about 10 birds that died then. And that was what the vets determined from tests. It was Pseudomonas and maybe E. coli. Now, those, if you have someone who dies of influenza in a human being, usually they're dying from pneumonia. They first get infected with influenza, but then you get secondary infection with the bacteria. The same is true for most of these animals and the chickens. Most of them are not actually dying from the influenza itself. They're dying from Pseudomonas, mostly, that then basically kills the birds. Next slide, please. So there was a report that there was dead birds on the farm, And the CFIA came in and they took 2 dead birds that had been on the ground for certainly more than half a day, maybe even almost a full day. They took a swab from basically the cochlea, the anus of the bird, and also from the throat, from the 2 different birds. And it went for a test. I think actually it was here in Abbotsford, maybe it was tested. And they got a positive result using the PCR test. And without getting into a lot of explanation, with a PCR test, this polymerase chain reaction test, it amplifies the amount of genetic material. So you have enough material that you can detect whether or not this is actually what you think it is. In this case, you're testing for the H5 protein, or gene, I should say. So they got a positive result, probably with more what we call 35 cycles. Now, just to put that in perspective, if you test for SARS-CoV-2 virus and you had to go more than 25 cycles, then what you're detecting is fragments of the virus or just straight false positives. At 35 cycles, you're about 90% false positives. So using this test, now in court, and the judge has even cited this in the documents, the claim was that on that date from that test, that it had H5N1 highly pathogenic influenza virus. That's not what they tested for. They only tested for H5 and H7, which are associated with avian virus, influenza viruses, and it doesn't mean that it's highly pathogenic. That was never tested. The N1 protein was not tested at the time. Within 41 minutes of that test result, the call order was immediately issued for the farm. Okay. And the result of that was that the farmers basically tried to fight this in court after they had consulted with a lot of other experts, including people that I know and myself. And so the call itself happened on November 6th through to November 7th. Okay, it didn't all happen like instantly. This was where according to the CFIA, Canadian Food Inspection Agency, there was 330 ostriches on site. They couldn't count them. So, I mean, sometimes there's like 313, sometimes it's like a little bit more than 330. These birds are running around all over the place. But they were herded into kill pens that were made out of bales of hay. And they were shot by an unidentified number of shooters, anything from probably 2 to 5 sharpshooters. Over 1,000 rounds were thought to be heard between that night, as soon as it got dark and the lights were shone out of the pens so that anybody watching could not really see what was going on inside. But the next morning, there was another round of shots because a lot of the birds, certainly some of them, had survived after being shot through the night. And then the bodies were loaded into trucks that were brought actually to, I think, Surrey, somewhere outside of, I think, one of the locations for Save on Foods. Super Save, thanks. Thank you. Super Save. And then we don't know what happened to them. They got buried into a landfill. Next slide. Now, the whole process of calling out the birds is to control the spread of the virus to other farms. Now, what's important to understand is that the birds themselves are usually kept in the farms. They're not usually transferred to another farm. And the virus can spread from a farm animal to a person, but it doesn't replicate very well in the person because the virus, like I said earlier, has evolved to infect birds, not mammals. It can do it, but it does it very poorly. So unless you have a farm worker that you've got a really big outbreak and that farm worker is now going to another farm, it's not likely to spread that easily. Now, if we look at the incidence, the scale up is the number of outbreak cases here in Canada, and this is over time since the first discovery of the virus. You can see it's pretty seasonally. Now, the stamping out policy, basically this idea that you kill all the birds in a zone or on a farm so it doesn't spread within the zone, apart from when we had this really almost double peak there, it has not been reduced by the culling stamping out policy. And it will continue to do this as long as the virus is spread in wild birds. You can't control it.
Duck migration.
Yeah, ducks and geese, which we cannot kill. Okay, next slide. So this CFI stamping out policy to control the spread has not really been working. I think that's the main point I want to make. And if any bird, if it's tested for H5 or H7, it's an immediate call. It's under very rare circumstances that, as we understand in court, there's maybe only been one case in the whole of Canada where they've actually granted an exemption. And that's because these are very valuable birds and they were housed in separate units. So they would kill one of the units, but they left the birds okay in the other unit because they didn't get a positive test in that unit. Normally, they would just kill all the birds on the farm. So this has resulted in the culling of about 17.3 million birds in the last few years in Canada, over 200 million birds in the United States. I think even the Food and Drug Administration with Kennedy, Robert F. Kennedy Jr.'s oversight, they realized that this is not working. And now they're getting it in the farms with the dairy. And the dairy, there's been over 1,000 dairy farms in the US, and they do not call the animals because it's just not practical. They usually isolate, allow it to recover if it can, and then it can go back. Now, the CFIA will be the first one to tell you that if you have meat that's been contaminated with H5N1, it's perfectly safe to eat, as long as you cook it. Most people cook their meat. And the milk, if it has H5N1, after it's been pasteurized, is perfectly safe for human consumption. Okay, next slide. That's good. So the question then is, why are they doing this? And this relates back to the World Organization for Animal Health. And as you may have also heard about the One Health initiative that's underway now with United Nations and the World Organization for Animal Health. The idea being that the environment, the animals, and people are all equally important in maintaining the health of the planet. And in principle, that's correct. However, how is this being enforced? And so, according to the World Organization for Animal Health guidelines, which is guidelines that are put together with input from various organizations like the Canadian Food Inspection Agency, which actually had a major representation in developing this stamping out policy in the first place. If you have an outbreak on a farm with poultry, with highly pathogenic influenza virus, now I'm talking about the World Organization for Animal Health rules here, then if you stamp out, if you kill basically all of the animals that are in that unit, then after 1 month, if there's no further outbreak in the zone, and we could be in the Lower Mainland here, where we have many different farms. So you lose the status if any one of those farms in the zone has it. But if no other farms have it, then you have disease-free status for regained after 1 month. It used to be 3 months, actually, when the virus was less deadly. But now, in the last few years with the current H5N1 strain, if you have this kind of an outbreak, then you only have to wait 1 month instead of 3 months. Yeah, I know it seems counterintuitive to me too. But if you let it burn out, if you do not cull the animals, but you let the virus take its natural course, the survivors, after 1 year with no outbreaks, then you regain the disease-free status. Now, the disease-free status is important for international trade of the animal products outside of Canada. The Universal Ostrich Farms was in its own zone. It was 10 months from disease-free in terms of showing any symptoms, 2 months away from having what would have been the disease-free status. It did not affect the trade, with a few exceptions. China and Mexico, which have very, very low percentage, well, 70% of our trade of poultry products is with the United States. And so they have these zones, but it doesn't matter what goes on in the country or even the province. Just a few countries where the province makes a difference. Okay, so the day after the call, 2 outbreaks of H5N1 occurred. So the day after the call at the Universal Ostrich Farms, and there was about over 40 cases in that time period. It would not have affected international trade at all. That's the point I want to make. Okay, next slide. So I've already outlined how to be highly pathogenic, it has to kill 75% or more, but yet these ostriches, it was only about 15% of the entire flock that actually died. And that's letting it go for, at least 11 months, basically since the virus first hit the flock. So it establishes that there was probably natural immunity in those birds and that they had antibodies. Next slide. So, and as a fact, I know they had antibodies because they actually, with our team at Connexus, tested it. Now, we could not test the birds after they had been infected officially with the outbreak in December of 2014. But we had eggs from the birds from the summer of 2024. And this way this test works, we have 40 different markers for the H5 protein and the N1 protein. So the more spots you see, the more different antibodies against the virus was in that bird. Each field is from a different bird. It's eggs. And our controls you can see on the side there. So these birds were probably infected with avian influenza virus in the years before, like probably a lot of the birds throughout. And they had antibody protection. And so if you have an antibody against the bird and your immune system is activated with your T cells as well, then when the virus comes into your body, your immune defenses rapidly take it out. You're not likely to be spreading the virus. Next slide. So the CFIA, after they issued the call order, in the Winnipeg National Microbiology Lab, had the full genome sequence of the specimens that they isolated from those 2 dead birds. And they determined that there's 8 separate segments for those 10 genes in influenza virus. And they found that 5 of those segments were actually from a highly pathogenic influenza, but 3 of the segments were from low pathogenicity virus, which could account for, in part, why we saw a very low lethality rate. The other is that, that's good enough. Next slide. So they didn't seem to have any sign of infection after January the 14th. All the birds apparently were healthy. They had no symptoms, but the claim from the Canadian Food Inspection Agency in the federal courts, and I was involved as an expert witness, basically for the farm, and I was privy to all of the information that was being provided by the experts from the CFIA, and I could evaluate them scientifically because of my own expertise. And they claim that the water in the soil at the Universal Ostrich Farm site would still have active virus. So these are healthy birds, but they're still shedding virus that could then infect wild birds that are coming to the site and then take that virus and then go to other farms and spread it. Ignoring the fact that the virus was already endemic in the wild birds. So then they said that, well, this reassortment of the influenza virus genes, this mixing of genes, that this was one they hadn't seen before. Well, what they brought out in court under cross-exam is there's been 100 cases at the time of the 500 cases of where they had actually done sequencing. 100 of them had different reassortments of high and low pathogenicity viral genes. So this was not unique. And it certainly illustrated that it was probably not highly pathogenic virus. And remember, the World Organization for Animal Health guidelines applies to highly pathogenic influenza virus, not low pathogenicity influenza virus. No, just go back. So then they said that the ostriches may serve as incubators for the virus that could cause a human pandemic now, okay? And serious illness, at least in people. Next slide. So I think this claim that the ostriches have no signs of infection, but they can still shed active virus, this goes against everything that we know in immunology. Like this, I found so incredible that scientists would be making these kind of statements that the active, healthy animal is likely still shedding virus. When you have influenza, and you all, at this age, you've all had influenza, the chances 2 weeks later that you're going to be shedding the virus after you recover, we don't take special precautions for that because it's unnecessary. You recovered because you made antibodies and your immune system was activated to eradicate the virus. The claim that the site could be contaminated for years, the judge said years, the actual experts said at least a year. This is based on taking lake water, inoculating it with virus, then filtering it so that anything that is larger than the virus would be removed, then putting it in a refrigerator at just about freezing temperatures in the dark, and then they could recover virus was preserved that way. However, when you have the virus on the ground, UV light from the sun destroys the genetic material. Microbes in the soil eat it up. So this is not a good comparison. And then you have to ask yourself, well, if they really felt this way, why, after they slaughtered all these ostriches, did they take the hay that was in the area with the blood soaked and spread it over the entire site? Right? Okay, next slide. And why don't they warn you that every time all these birds are flying overhead and landing on your fields and your parks and the schoolyards, they don't tell you, oh, better watch out. This is the migration. They're all full of H5N1 and we're going to spread it to everybody, right? You have to put it in perspective. So as I said, there's already been these reassortments described, and historically, over the course where this has been tracked, the number of cases in the last 5 years with this so-called very deadly H5N1 strain, there's only been about 71 cases in North America. And of those, we've had 2 deaths. 2 elderly men, 65 years of age, had chickens in their backyard. They had comorbidities. Okay, this is not a very, very lethal disease in humans. But in the courts, they talked about how historically they've gone back with pretty close to about 600 cases in total around the world, and that 48% of them died. But most of those cases are in Vietnam and in Cambodia and China, in farms under very poor conditions. And we can see with this is not a highly lethal virus in people. In fact, most of the people of those 70 cases that are diagnosed, most of them were on people that were involved on dairy operations, not poultry. In fact, there's about 50% more cases are from dairy farms than the poultry farms. And most of them had no symptoms. They were just tested by PCR and found to be positive. Okay, I think that's the last slide. So thank you very much for your attention. And